want to, oh, as my colleague has just said, if you don't want to be recorded, then please will you switch your camera off, but obviously don't leave the session, but be aware that this session is recorded. So the format of the seminar will be, I'll invite Wilfred. I'll give a short introduction to Wilfred for those of you that don't know him. And then he will speak for about 45 minutes or so, and then we have a session for questions and answers at the end. So please feel free to add any questions or comments or challenges in the chat, and then I'm sure Wilfred will sweep them up at the end. So thank you again for everyone for joining us this afternoon for what I think will be a really, really interesting seminar. So as I say, I have great pleasure in introducing my colleague who's sitting across the table from me. So we're in the same room, but we can't share the same screen. But anyway, so Professor Wilfred Otten joined Cranfield University in May 2016 as Professor of Soil Biophysics. Before that, he worked for 9 years at Abertay University in Dundee as Chair of Biophysics of Soil Ecosystems, and he was also co-director of the Scottish Informatics, Mathematics, Biology and Statistics Centre, where he developed innovative methods to characterise soil structure, and I'm sure we'll hear much of that research this afternoon. Prior to that, Wilfred worked for 16 years at the epidemiology, I knew I would have problems with that word, epidemiology and modelling group of the Department of Plant Sciences at Cambridge University, working on invasion and persistence of soil-borne fungal plant diseases. Wilfred has a degree in agricultural engineering with special reference to soil science and fertiliser use from Wageningen University in the Netherlands. And his PhD was in horticultural sciences, where he focused on closed fertigation systems, also from Wageningen. He was president of the British Society of Soil Science for between 2017 and 2018. Wilfred's general research interest, as we'll hear this afternoon, is in physical, biological and chemical processes in soil. His current and past research has equipped him with multidisciplinary expertise in soil physics, chemistry, microbiology, and mathematical modelling. He's an expert in the characterisation of microhabitats in soil. He's used research techniques such as transparent soil, X-ray CT to characterise internal pore geometries and plant growth, root growth, and I know again we'll hear much about that this afternoon. He applies his expertise to develop a quantitative, integrative and predictive approach that forwards our understanding of soil processes to enable sustainable management. So it gives me great pleasure to hand over to Professor Wilfred Otten to give the seminar this afternoon. Thank you, Wilfred.
Can you hear me now?
Yes, thumbs up.
Yep, thumbs up. Okay, good. So thank you very much, Jane, for the kind introduction. And it is an absolute pleasure to be here at this seminar series and to give a talk today about fungal invasions in soil. So what you see on the background is what I call my pet fungus. So that's Rhizoctonia solani, and it looks like a very pretty picture. This is taken on an agar plate where very often in the lab fungi are cultured. This is a low nutrient one to make it more realistic. However, what I will show today is that it's not very meaningful to look at fungi on agar plates, and that what happens in soil is going to be completely different. So what you do see here is an important characteristics of the fungi, namely that they spread. So one of the messages I'll get across is the importance of spreading of organisms through soil, how that determines the way they function, but also how that gives opportunities for us to control their spread should we wish to do so. Then I'll briefly go over some emergent technologies that will enable us to look inside soil at scales that are relevant for these organisms to operate. So very often we look at soil with our own eyes, but that's at a different scale than when organisms experience it. I will then move on and show how some changes in the environment can result to tipping points or thresholds that particularly relevant, of course, that if we make minor changes to our system, that all of a sudden a fungus can invade. When it's a pathogen, it can bring huge yield reductions. And then I'll ask the question, what would be the key driver to soil fungal in soil? So to soil fungal growth in soil. So those are the 4 aspects that I will go through in this presentation, and we'll see how far we get in 45 minutes. So, one moment. I need to move on the slides. Okay, so this is just a simple first introduction where I wanted to point out and clearly demonstrate the importance of soil and the difference of spread on the surface or spread through soil. So what you see here are 2 curves. The one on the top here is when we put a surface on the top of a soil and follow how it spreads. And this is when we place it inside the soil and we follow how it spreads. Now, it's not surprisingly that the spread through soil is less than on the surface, as it has to find its way through a tortuous network of pores with many obstructions. But what is surprising to see here is that this completely different rate of spread and extent of spread happens at the same biomass. So biomass dynamics and the biomass growth over time is identical for those 2 colonies, but one spreads a lot faster and further than the other one. So this is quite important because very often when we quantify fungi, we tend to look at biomass or colony forming units or whatever measure we quantify them at. And very often we do not look at dispersal and the rate of spread of organisms. Yet it is the rate of spread of organisms and the way they explore soils that is the most critical characteristics to observe. So I'll start by considering space as a result of that. So the fungi will explore the space. So how does that matter? And I'll start with an example of damping off epidemics. And then I'll move on to a bit of theory in there, which is called percolation theory. And I'll use that to demonstrate how thresholds for invasion can exist and can be controlled. After that, I'll move on to extend these ideas for epidemiology to invasion through soil and carbon dynamics. When we look at an epidemic, and here what is going to look on the right as a bit of a clumsy simulation is actually observations that we took over time. So you have on the left a population of young radish plants, and Rhizoctonia solana will spread from one plant to the next and will cause a damping off disease of young seedlings. On the right, the red dots is where we placed some inoculum in the soil. And what will happen next is that you'll see that in first instance, you'll get some primary infections. And from those primary infections, it starts spreading to neighboring plants. So you'll see that ultimately the epidemic is predominantly dominated by spread from one plant to the next plant, rather than, in this case, the amount of inoculum that was left in the soil. So this is roughly how epidemics will progress for so many soil-borne diseases. The way we very often quantify them is by looking at disease progress curves. So you see 2 examples here of a disease progress curve, which basically summarizes the cumulative number of disease plants over time. The dark lines are averages of the replications that you see in here. I think this was replicated 13 times. So you see how variable epidemics can be, which we very often observe. But you also see that there's a clear difference between the 2, so a clear effect. And I'll come back to what's causing that in a moment. But looking at the picture, if this had been a treatment of control, you would say that there has been some reduction as a result of the treatment. So the conventional analysis compare disease progress curves. We can look at the final level of disease. We can look at a delay in the onset of disease. So there's various ways by which we can compare those curves, and that gives us a quantitative measure of the control, like did it work, yes or no, but it does not give us insights into how the control took place. Now, does that matter? And I'll show you why that would matter. So no doubt, we still have got COVID in our mind, and we know how important it was when there is somebody diseased that you spread it to a neighbor or a colleague or somebody else. And very often that is determined by the contact that we have. In plant epidemics are not different, but the contact is more regular because plants don't move, unlike humans do in a population. But the epidemiological models, I struggle with it as well, are similar. And here I give a very simple example just to demonstrate how we could do this empirically. But effectively, you see here infected plants, they are new infections, and they have a number of contacts with neighboring plants, and then we can follow over time and calculate what the probability is that a neighboring plant became infected. So that can be translated into what's known as a transmission rate of infection upon contact. So therefore, the epidemic is driven by the number of contacts, and of course, as the epidemic increases in size, the number of contacts is increasing, and also the transmission rate between the two of them. Importantly, we can, by simply counting, calculate those transmission rates here, but obviously more sophisticated models can also be developed, and we've done that, but this is just for demonstration, a more simple representation of how this works. So what does a typical transmission rate for those damping off diseases look like? You get typically a bit what looks like a rise and fall dynamics as plants grow and resistance increases towards damping off. And this will determine the rate of spread that you saw in that first video, when you start with the initial infection and then the rate of spread and then the epidemic stops. So this is the race of rapidly spreading pathogen versus increasing resistance of the host as the host plant becomes stronger. Now, what I've done here is that for the two epidemics where we probably would have said that there was an effect of the treatment, I've calculated the transmission rates on the right. And as you will see, the transmission rates for the two epidemics are identical. So if this has been a treatment, the treatment had no effect on the rate of transmission between the epidemics. The only reason why those two epidemics were different is because there was a lower number of infections left from the previous season in one of the curves, in the lower curve versus the higher curve. And I know that's true because I did the experiment and I designed it that way. But the importance here is that if we would have just looked at time series, we would have concluded that our treatment, whatever that was, had an effect on reducing the epidemic. However, if we followed more a mechanistic process, we would have seen that actually there wasn't effect, but this was purely determined by the amount of disease left from the previous season. So the wrong conclusions might be drawn without considering spatial aspects. So this is one of the reasons why space is so important, not just for the fungus to explore the environment, but also for us to consider this in how we analyze what's happening in the environment. So I'll take that a bit further now, and I'm going to look at what is known as percolation theory. But again, don't worry about it. It's called theory, but we won't take it to equations or anything like that. So this will be, again, a visual demonstration of it. So in first instance, we look at the environment as discrete units. So that can be crops in the field, which are very often on some sort of a lattice or in rows, as is shown here in the field. We can also consider roots, where spread will happen from one root to the next root, and it is the spatial organization of the root system, which basically forms a lattice between which fungi will have to spread to cause an epidemic. So again, it is primary infection will be on a single root, but an epidemic will require spread to neighboring roots to kill off larger aspect of it. We can also consider favorable patches in soil, and I put it up here, but I will come back to that in a later part of the talk. The point here is that there's various examples where it is convenient to visualize spread through a population of discrete sites on a lattice. So if we consider the environment in such a way, we can look at how fungal hyphae will grow. So we'll have a donor site, which can be a spore, which correlates and starts developing fungal hyphae, as you see here. Then it will grow and meet a recipient, either a recipient host or a fragment of a food source, carbon, which it will colonize. And the degree at which this colonization will happen depends on the distance. So if there's only a little distance, so a high plant density, you'll have a very high chance that the neighbor gets infected. If they are further away, that distance, that probability becomes less. That's known as a dispersal kernel, or for ecological organisms, or pathozone for epidemiological systems. Now, very often people focus on how far an organism can spread, but that's actually not that relevant, because if we move on from a neighboring host to a population, as you would see here, then the theory of percolation will predict that there is a threshold value, a threshold distance or density, or a threshold probability that is required to switch from non-invasive spread to invasive spread. So that's a non-linearity in our response. It doesn't matter how far something spreads, it matters how likely it will spread a certain distance. So very often people focus on how far it spreads, but we should focus on how likely it is that it will colonize a neighboring plant. Now, this of course is theory, so as we do, we set ourselves out to test this. What you see here on the left is a simulation just to show how sharp this threshold is. It's a real threshold. So if you move from 40% of your sites protected, either by chemical protection or resistant varieties or whatever, then you have still invasive spread through your entire system. If you had 42% removed, so a very minor change, then your epidemic is controlled locally. If we do this empirically, so the threshold would be at 0.41 roughly, and that would be a theoretical threshold in there. The empirical system where we tested this shows a non-linearity. So our theoreticians would say that doesn't prove anything, there's not a threshold here. But what this shows to me, and what I think is important in here, is the sharp non-linear response to an environmental change. So that means that when we observe our systems, and we are in this region here, we can make quite big changes to the environment and think that our factor has no impact on controlling the disease. Sorry, just lost my cursor. But if we move a little bit further, then we have a very sharp non-linear response. So if this is something which we want to invade, then obviously we think here we are safe, but then all of a sudden the system collapses and we lose either our beneficial fungi, or if it's the other way around, we'll get an invasion. So minor changes in the environment, in the system, and note that these changes can be either introduced by a density of crops, so it suggests there's a critical density of crops, but it can also be changed by wetness in the soil, which we'll come to in a moment, or even soil structure. So these are some of the theories which are known in epidemiology and well established, and it has always struck me that epidemiology is fairly well understood and has got fairly well developed models, and it was never clear to me why we haven't got the same approach and the same understanding to soil biology. When we move to soil biology, when we move underground, we seem to forget the ecological or epidemiological understanding that we really would want to have below ground as well, to make sure that we end up with a predictive systems. So we try to look at, can we extend this, for instance, to what happens below ground? Can we extend this, for example, to have a more predictive understanding of carbon dynamics? Well, the easy thing is that, of course, we think that it's far more difficult to do on soil. And here you say that dispersal kernel I showed you before which was on a 2-day surface, but now we have quantified this in a 3D surface. So this is experimental results. So it just shows you that although doing this in soil is far more complicated, but we can actually get an idea of how well the fungus will spread through soil for specific conditions that we have imposed. So what you see here is distance to the right, and where you have a decline with the distance, as we saw before, and going backwards, you have an increase in time. So initially there's a low probability and it goes back on time. Now, what you should see is two sigmoidal curves, one going down that way and the other one going up, going backwards. If you don't see that, just blink your eyes, because it's your brain that can reverse these graphs very easily. But this gives us an opportunity here to characterize the complete dynamical growth of a fungus with only 4 parameters. So a relatively powerful and very simplistic system, but this enables us to quantify the impact of soil, soil wetness, soil structure. And the things we look at, if a structure is more variable, more heterogeneous, we will see that we get a more sharper decline and then a more longer tail at the end of it. So, and when the soil gets wetter, it will be more compact. So this is the quantitative information, which means that the sort of ideas I spoke about earlier could, in theory, be done in soil. We can quantify fungi in the same way. We asked ourselves, what are the pores that fungi grow through? So we wanted to know, when pores are air-filled or water-filled, which ones do they prefer? Do they spread at all through water-filled pores, or is it mostly the air-filled pore volumes that matter? So in order to test that, we looked at air permeability in first instance. So if a sample is saturated, you try to squeeze air through it. It doesn't work because all the pores are water-filled. When you dry your sample out, then gradually there is a point where there's a connective network from one side of the sample to your next one, through which air can flow. Again, this is a sharp transition. So this is the point where a connected air-filled network exists when you start drying your soil out. How does the fungus respond to that? Well, the colony radius responded in the same way. So we can see that when we have fungi in a wet soil, it does spread through wet soil. So it's not that it can't grow through a wet soil, it does. But then what happens is that when the soil dries out and we have that connected network, is that a rapid increase in spread occurs. So a clear indication that there is a response to air-filled pore volume for fungi. They have a preference for following air-filled pores and spread more rapidly. We also quantified the biomass in there, and you see actually the opposite happening. So this gives us an idea that when we have a bit of a constrained pore space and a poorly connected air-filled pore volume, that a fungus will form very small and dense colonies, but it then switches rapidly to a well-connected network, to larger colonies with a lower biomass density. Now, obviously, both are important, the density and the spread, and they can both have different impacts. If you've got a lot of local nutrients, you may want a denser colony, but if you have sparse nutrients that need to be explored, you won't have this rapid advance. So it's well known from fungi that they can switch this sort of behavior from exploring to exploiting the environment in relation to nutrient availability. But here we showed that physical properties can have the same effect. The importance again is that you saw how rapid this change can be. So here too, we have that physical conditions can introduce a rapid switch in behavior of a fungal colony. So you may find that your fungus is not invading, but then a minor change can happen in your environmental conditions, and it results in a rapid spread of your fungal. And if this is a pathogen, you have all of a sudden a rapid spread of your epidemic. This is just a visual presentation. So what we did here is what they call microfluids. So these are sort of transparent media where we can visualize what I just described. And actually, when they did this experiment, they set up this experiment to prove I was wrong. But unfortunately for them, they found that it was right. So you've got solids here, you've got water-filled pores, and you've got air-filled pores. And what you see here is that this medium is printed and prepared and based upon an X-ray CT scan we did. So it's sort of a transect that we've taken through a soil volume, and they put inoculum and let the fungus spread through it. And as you can see, it does spread through the water-filled pores, but it spreads at a much higher density through the air-filled pores. So this is what it really looks like in soil. And at the moment, the best way to look at fungi in soil is still in 3D. And if you remember the picture of the opening slide where we saw this very nice network, this is the same fungus, but this is when we look at it in soil. So what you see here is what we could refer to probably as an aggregate, and an aggregate up here, and here we have some pore space between the aggregates, and we can see how the fungus will follow this as almost following a highway through soil and preferentially spreading through these larger pores. So be aware that what we look at is actually a very high density of hyphae, because this is very close to a very potent nutrient source that I put into the soil to be able to visualize it in this way. Now you will agree that this is a completely different picture than we saw at the opening slide, where it is the fungi that determines how it grows. But here we get a different picture of a fungus that has to find its way through a complex networks of pores in soil. It also shows that what we often do is taking soil apart and sieving it and looking at aggregates, that when it comes down to fungal spread, those aggregates are not that important. It's actually more how things are being put together and looking at the pores that are explored in soil. That is what matters. So the action is in the pores rather than in the solids. Now that is a look at how can we look at that, and I'm sure most of you will have heard of X-ray CT, but if you haven't, we have put an example here together of what an X-ray CT image looks like. So this is a cubic centimeter soil roughly, and what you get out of that is a grayscale, and I will let it run as a video. So in that grayscale value, which is basically looking at different densities, we need to figure out what are pores and what are solids. So what you see here now is what are the pores that are connected to the outside of that cube. And in the moment, we take the solid phase away, so we take the brown stuff away, and what will be left is the pores that are inside soil. So here you will see in a moment, for the first time for some of you, what soil looks like on the inside. Now, when I studied soil, I was told that soils is a bundle of capillary tubes, and you can see here that it's far from that. It is a very complex geometry through which organisms will have to spread, through which water will move, through which nutrient diffuses. The change of color for this example is just which pore space is connected with each other and what is disconnected. Okay, so within this pore space, if you keep in mind, we just took a 2D section through that, which you'll see here, of a loosely packed soil, and you will see the black is the solid, so we thresholded this so we can distinguish between the pore space and the solid phase. Then we looked at where the fungi are under the microscope, so we went through this section and looked at every little square here in this section and identified where the fungal hyphae were in there or not. And the black dots that are on here are the microscopic sections within this larger section that we looked at and where we found some fungal hyphae. So then we overlay those two so you can get an idea about where fungal spread has occurred within this soil. And we can see roughly again that we find most of our fungal hyphae in this sort of lighter area, so more porous and less dense area. I always say fungal mycelium, it's a bit of a lazy organism, and finds the easy way out. When we consider how bulk density will affect that, on the top here you see what bulk density is doing to your pore space. So typically you'll see that if the density increases, the larger pores get more compressed, and basically your other sections, your solid phase gets fragmented, so you get many small pores in here. And at the bottom you see how the fungus is responding to that. Now, intriguingly, although at a high bulk density, it looks far more difficult for the fungus to go through there, you will see that the soil is very well colonized here. So that's, in first instance, counterintuitive. And at the low density, you will still see that it follows those highways. Now, do remember, this is about 2.5 centimeters or 3 centimeters, and a fungal colony can be larger than that. So we basically look at the center of a fungal colony. So it's likely that when we go further away at a high bulk density, it drops off, whereas at a low bulk density, it will spread a lot further. But a low bulk density, sparse colonies following preferential pathways, a high bulk density, dense colonies entering smaller pore spaces. So the small pore spaces that are entered here are ignored here. Now that's interesting phenomena, and you have to ask yourself, why is this happening? But also, by simply looking at those pictures, you can already imagine the consequences. If this was a fungal pathogen looking for roots, then it's a very high probability that it will find a root. If it's at a lower bulk density, it will spread a lot further, but it is less likely to encounter a root. Although, of course, we'll have to consider that roots have a similar behavior and also follow the easy pathways through soil. So maybe there's that to consider as well. Equally so, if this was carbon that needed to be accessed, there's a far more intense connection with soil carbon than there would be here. Or when it's a mycorrhizal fungi, you'll have a better colonization of your soil over a shorter distance or over a longer distance with a less intense colonization. So it gives you immediately an idea that these sort of things and the way soil structure will control your fungal growth will have quite big consequences for the way soil functions, or for at least for processes that involve fungal hyphae. Now, although I said the fungus spread through the air-filled pore space, if you look at the 2D section, I had inoculated this in the center, and somehow it has spread through the areas it doesn't like and ends up in those large pore spaces. But equally so, there's other, which you would expect to be well accessible pore spaces that remain uncolonized. So how does this happen? What happens? And this is one of the restrictions we have with thin sections. We can't see how these areas are connected with each other. So thin sections are still the best way to look at biology in soil at small scales, but they do not tell us how those sections are connected with each other. What we did is that we combined the two together, but I'll first explain you a little bit what connectivity looks like. So here we have the pore space lifted out, which is the connected pore space. And here on the right, you will see what a section is starting from the top, with the gray area that would be the connected pore space and the white area that would be the not connected pore space. Now I challenge you, just put your eye on one spot in the connected pore space, and you'll still see that when this moves down, how quickly this gives you a dead end. So although this pore space is connected, it is not connected in a straight line from going from the top to below, but it is a very tortuous pathways that need to be explored by the fungus to find its way through this soil sample. So you can immediately see that this will have a big impact on the spread of the organisms, which we saw in the first slide that I presented. Okay. Of course, it doesn't only matter what the pores are. As we know, pores are either filled with water or with air. So we would like to know where the water is within those pores. Now, we can see for some circumstances where water is inside the pores for very small samples, either in a synchrotron or for some of the lighter soils where we may be sometimes lucky enough that we're able to see water inside the pores. But for most situations, we're unable to see that and unable to distinguish it from air in the pore space with X-ray CT. Now here, modeling can offer an outcome. So you'll see here two examples of a very small part in soil. The one on the left is where we went to a synchrotron and get very precise scans where you have the solid phase and you have your water-filled pores here. And on the right, you have a modeling output of a scenario which was done by colleagues in France. And you'll see that this gives a fairly good agreement. So this is what's called Lapis-Boltzmann modeling. So it's based on particles bouncing off the wall and coming together and determining where water is within this volume. Now this gives us some confidence that although we can't see water, when we put some physical models in there, we are fairly well able to predict where water is inside the medium. Organic matter is another thing, so I'm just going through a few things that we can see. It is very challenging to see organic matter, but particular organic matter we can see within the X-ray CT scanner. So here you see what particulate organic matter distribution is inside a small aggregate, which was done by colleagues at Michigan State University, and they developed computer learning programs to segment out the organic matter. Now this is particulate organic matter and fragments of roots that you will see inside an aggregate. Of course, there's also organic matter which is a bit more diffuse spread through this aggregate, and that's very difficult to distinguish with X-ray CT, but we can get to that if we use something which is known as staining with osmium, and then we can also visualize where the rest of the organic matter is. So this is still an area of development, but we are beginning to get an understanding of where the pores are, where the water is, where the organic matter is, and also where the organisms are. So we're beginning to get a complete picture of what soils looks like on the inside. Now these are nice pictures, and it's always good to develop technology to get an idea what things look like at these very small scales, but it doesn't really give us the understanding, and that's still where we have a challenge. So over the last years, the development of technology has progressed enormously, and we are now able to see things inside soil undisturbed, which we wouldn't think would be possible a few years ago. But of course, seeing is still a snapshot in time, and it doesn't give us too much insight about what is happening. So this is where we've moved on to use modeling approaches to consider how fungi will respond to these environments and how this will impact on the functions they perform. So I'll give a brief overview of there, but don't worry, I won't show any of the equations because I know they can be challenging, but it's probably important to point out that the equations that are in there are fairly standard. So Michaelis-Menten equations for uptake, but the characteristics of it is that we in the modeling consider fungi as a network. So we've got a network here, we have growing tips, which are we call non-insulated biomass. So it propagates or diffuses through your porous space. Fungi typically grow at growing tips. Then we have got insulated biomass, which is the sort of center of a fungal colony, which retains as hollow tubes before it dies, and a lot of internal resources can be transported through there. So you've got internal resources propagating through the mycelium. So characteristically of fungi is that it can take up carbon at this end, transport it through the mycelium, and result into growth at the other end. So a fungal colony, I always say, integrates its environment and integrates the heterogeneity that you will find in the environment. It also characteristically can die in the center. So old parts of the mycelium can die, and other parts of the fungal colony can take up those nutrients that are released and use it to keep on growing. So it's a sort of a almost infinite growing system. We have particulate organic matter, which need to be degraded by enzymes and becomes dissolved organic matter, which then becomes available for uptake and has got the distributions between the different fungal aspects, whether it is recalcitrant or growing, and we have respiration through CO2. So these are some of the characteristics that we put in the model. And what we ask ourselves is what happens when we put something like this in the environment. So a couple of simple results I will show. So first we looked at what happens when we simulate fungal growth through these different structures. And on the left, you see something that has been a native succession. And on the right, we had a corn, soybean, wheat rotation in the US with no tillage. Now, we can always ask, what would be a good structure? What does it look like? Here you see two images of two completely contrasting pore structures, but both are perfectly well functioning ecosystems. So what a good structure looks like is not easy to answer and not easy to get from your quantitative measurements. But here you see the largest connected pore cluster in two contrasting management systems. The connectivity changes with the different tillage systems we had, so native succession, conventional tillage or no tillage. And you see typically that the more fragmented pore space in the no-till treatment will hinder invasion. So we had on the left here some fungal invasions in there, typically showing how a fungus in a soil will spread through the pore volume and explore this. The larger connected pores of the native succession and conventional tillage promote invasion. So we are able to make some sort of a relationship with long-term field trials and get some predictions out there about what the consequences are of different managements for fungal growth. But there's still a lot of things that we don't know. So one of the things that modeling can do is the what if scenarios. So at some point we'll have to accept that at this moment in time we can't quantify everything. So we have a reasonable idea that we can get at particulate organic matter, but for larger samples we're not able to do that. But we can look at what the distribution looks like and put similar distributions within our pore space obtained from X-ray CT. So with an X-ray CT sample, it gives solid and pore space. We can make a distribution of organic carbon in there, which will dissolve in dissolved organic carbon and diffuses through our medium. And we can look at what different densities and different distributions the impact would be. So when we look at that, we see again a nonlinear response. So we here changed the distribution or the amount of soil carbon, and on the top you just see what it would look like from very low densities to higher density of soil carbon distributed within that soil volume. As you increase soil carbon and you put fungal in the model in there and you let it grow, you will see that for very low carbon contents it grows a little bit, but then it stops growing. So it cannot access any further carbon fast enough, and it will start degrading and it will not invade your soil sample. At higher concentrations, you see that you'll have invasive spread, and the higher the concentration, the more spread we have. Now, all the carbon that we talk about here is in principle available. The switch is approximately at 3% carbon, but be aware that that's not necessarily a realistic number because it depends on how we parameterize our system. But what this shows, again, is that in response to the amount of carbon, our fungi will have a nonlinear response. So again, it is this nonlinear change of fungal behavior in response to environmental conditions. It may not be exactly at 3%, but this nonlinear change is generic, and we would find that every time. That is something which currently is not in any of the soil carbon models. They will not consider this nonlinear response. So these are some important novel insights that get obtained from these microscopic properties. There's another one where we can look at how do we distribute the same amount of carbon. So if the carbon is more clustered in larger particular organic matter fragments, or it is in very fine fragments and more uniformly distributed, we will see that that again has an impact on the amount of CO2 reproduced, produced or respired from the soil. So importantly, if you would consider it the other way, this is the same soil sample with the same carbon content as we would measure our carbon content, but it has completely different rates of respirations and completely and very large variability in there, which we very often observe when we take measurements in the field. This is a variable that can be very dynamic and very heterogeneous in time. But it also shows one other thing. We always measure on our samples the carbon content. Measuring carbon content is not enough. You cannot relate carbon content to microbial activity or CO2 evolution. It shows you that at the very least, we need to know a bit more. Even if we would measure some particulate organic matter distribution in there, which is not impossible to do, we would gain far more insight in how the soil would function. Some of the results are a bit counterintuitive and have nonlinear responses, but our models will explain this. So again, this would be one of the benefits of the models, but I won't go in too much detail at the moment. The important thing is that what we learn from the modeling here is that measuring bulk properties in soil is insufficient to understand the implications for microbial processes. So one thing we'd like to ask ourselves, and we always get asked, is what are the key drivers? We always want to know what is the most important thing that determines soil carbon dynamics. That's probably inherent in most of academics, that we want to know what is it? Is it biological? Is it physical? Or is it chemical? So is it the organic matter? Is it the physical structure? Or is it the microbial dynamics? So we tried with our model to see if we could answer this question. So here you have your physical environment, which is the structural look at the pore distributions. Here you see a simulation of fungal growth through the pores, and here you see the distribution of water through there, just as an example how we can bring this all together in the model. So how do we go about answering that? Well, first we wondered, if we have consider our fungal system, what are the important aspects we look at? So we've already seen that, of course, the pore space need to be connected. If it's not connected, you can't have any spread. And we've seen from experimental results that the connected air phase was also important. So preferentially followed by fungi, so higher rate of red. So you would expect that connectivity of air-filled pores is an important factor to look at. But it did spread through the water phase. And one thing that water phase does is diffusion of dissolved organic carbon. And if you remember, the dissolved organic carbon is required for uptake and also diffusion of enzymes to degrade the particulate organic matter. So a connected water phase from that point of view would also be beneficial for growth. Then we have the distribution of our particulate organic matter or our organic matter, which we can say if it's more clustered, it's disconnected. If it's homogeneously distributed, it would be better connected. So it's beneficial for a certain type of fungi. And then we have the spread and translocation. So the fungus itself also introduces a form of connectivity, as it is able to translocate carbon accessed at one site and transport it internally to another site where less carbon is available. So there are biological aspects for connectivity, chemical aspects of connectivity, and physical aspects of connectivity. And they all work together and have determined the outcome. So I'll show you first just a couple of examples of the environment, but then a few simulations that we run, not too many of that, just to demonstrate what the importance is of considering these different forms of connectivity. So here we have just a transect of pores, where in one we fill 20% of the pores with water, and then you have an air-filled connected. And if we fill 80% of the pores, you have your waters connected, but your air is disconnected. So just to point out that with simulations, we can create many scenarios to answer these type of questions. We picked 20 and 80%, also keeping in mind that the next stage where we want to experimentally validate these situations, we can create these sort of situations experimentally. So it's always that we kept in mind scenarios, what can we model and how can we validate that subsequently if hypotheses are formulated from our model. Resource connectivity, carbon can be anywhere, carbon can be spatially distributed. I already explained that before, but we refer to that subsequently as a resource connectivity. So very well connected carbon or resources, disconnected carbon or resources. Obviously, various degrees of this in between. Then we have our fungal or biological connectivity, if you would call that, and we just looked at what is referred to as R and K strategist. So you either have a strategist which is long-lived but has a slow growth and typically explores a nutrient-rich environment, or we have short-lived and rapidly growing organisms which will explore the environment. This just means that we put some different parameters in our fungal growth model, looking at different rates of spread based on traits that fungi have. So we introduce some fungal traits in our modeling scenarios to explore how different traits of fungi will respond to environmental conditions. Now, typical output of simulation when we introduce in there, and it will be sufficient here to just focus on the colors. So we'll have some time, so evolution of CO2 over time as the simulation progresses. And then we have different colors for unconnected resources and water phase, unconnected resources and connected water, connected resource, unconnected water, and connected resource and water. Now, I don't expect you to remember any of this. So for the next ones, I would suggest just focus on the colors, because what's going to matter here, and basically when we said what is important, it will mean that one of those colors should always be on top if that is a key driver. Now, what we're going to see is that we have all those scenarios, different structures in here, different water content, different resources, and different fungi, R and K strategies. Now, for the higher bulk density, we just found that the rate of fungal growth is faster in a high bulk density than in a low bulk density. And that was true irrespective of whether the water or the resource or the fungi is connected. We would always find this as a result. If we look at the bulk density and we look at an R strategist, then we'll see that the R strategist is spreading faster than the K strategist. So that would be what we find for a low bulk density soil. And of course, we want those results to be consistent, but we'll see when we change our physical properties that we will no longer find that distinction. So what we have started off with is effectively two different fungi that grow and spread differently on an agar plate, and we would have classified as different fungi. But when we put them in soil, this can be completely reversed. So the observations and the classifications of how we have learned how our fungi spread in the environment are going to be dominated to some extent by our environmental conditions and not just by the fungal behaviors or the fungal genetics. So, sorry, let's go back. So we very often want to know, as I said, what's the key driver? Well, unfortunately, and I only showed you a couple of those simulations, you already saw contradictions happening in there, and the colors will rapidly swap over from one environmental condition to the other. So it was not possible to identify a key driver. We wanted to know if it's mainly the physics or the biology or the carbon content that is driving the response, and we couldn't find an answer for that was universal. So for all scenarios that we explored, the physical connectivity was always important. The connectivity of organic matter was always important, and the biological connectivity was always important. So all of them had a role to play in how you explore your environment. But of far greater interest were the interactions. they indicate that one connectivity, if that gets a bit less, it can be compensated by another one. So if there's less connectivity in our carbon content, but we have a fungus that is very well capable of translocating, then the translocation of carbon compensates for the lack of connectivity in organic matter. Basically, it can grow through an area of soil where there's less carbon and wait to explore and find another area where carbon can be found. So important for physically well-connected pore volume is the water content. So, but only for well-connected pore volumes and not for other situations. So we get some indication about what is important, but we can't identify a key driver. And the other thing we identified that actually it is not the properties that we focus on, but it is the interaction that determines how a soil functions. And this is also an important situation for soil health. We always want to know what property do we need to measure, but it is actually the way things interact with each other that matters in soil. And unfortunately, interactions between properties is not something that we focused on and know how to measure. So a few points what we've learned from this so far. So attribution of traits to fungi depends on the environment. So we can think of fungi as growing differently on plates where we can observe them, but as soon as we put them in the soil, that behavior can switch over. So this is important to know that the environment is as much determining how fungi spread as the fungal mycelium itself. So as a result, you can turn this around. Selective pressures can be expected to be mediated by physical conditions. So if we have a diverse population of fungi, it means that a change in the physical properties will select one fungi over another and can, as such, support biodiversity. This is probably also why we find so much biodiversity in soil, because it's heterogeneous at so many scales. Small changes in the environment can invoke rapid changes in fungal pollination, so there's risk of tipping points. These are some of the earlier results I showed you experimentally and mathematically, and of course tipping points are very worrying. But I've also shown that there may be some reservations to be made against that, because those tipping points and those switches may exist in some situation, but they were still relatively simple systems as we looked at that. So multiple pathways of connectivity contribute to the outcome of the soil function. So connectivity matters. Soil study, this means that we should study soil as intact systems. So taking soils apart and sieving soil and analyzing the properties of fragments of soil is the same, we always say, as blowing up a building and picking a pile from the rubble and trying to understand what function it had within that building. So it is almost impossible to do so. And we need to rethink what we call connectivity for microbial mediated processes. So at the moment, even with those novel techniques that we now have, the focus has been on connectivity of pore space as being an important factor. But we've shown here that connectivity means a lot more, and it's a lot more complex. Now, is it a problem that it's complex? I think it's not, because the different pathways compensate for each other, and that will enhance stability of the function. So if one property drops a bit and would on its own create a tipping point, another process takes over in soil and will make sure that the function can still be maintained. So the complexity that we find in soil, and I spoke about, is actually what makes it function and what enhances stability of soil. So the expectation is that this impacts on resilience of soils to perturbations. And that, of course, is one of the most important properties that soils will have. We always want to know, how does it respond to sudden droughts, to flooding, and other perturbations that will happen during season. So that, I think, is my last slide. And I'm sorry this was a bit complex, but we always say it may be complex, but we always have to keep in mind that on the road to success, there are no shortcuts. So thank you very much for listening, and happy to take any questions. I think I've switched off.
Wow, thank you so much, Wilfred. That was just mind-blowing, literally. The content and the coverage of that was just amazing. And you've got some claps. Thank you very much. That was very, very interesting. And as I say, the journey that you took us on was fascinating. Do we have any questions? I have one question from Samir. Thank you very much, Samir, who says, thank you for this presentation. What is the effect of soil pH on fungal growth?
Okay, I hope. I'm just waiting for Jane to switch the volume off, but otherwise we're gonna get an echo in here. So yes, pH is one of the most important factors for a lot of microorganisms. We haven't looked at it specifically here, so I can't tell you how that will have an impact in the simulations, because we haven't included that yet. We decided to focus on the physical and the carbon aspect of it. But certainly in the rhizosphere, obviously, we have changes in pH at microscopic scale. So I can't say that I've looked at that yet, at the importance of variability of pH at these sort of scales, and we know it will happen. And I'm not fully aware how fungi will respond to that. I know in the literature that people have looked at how it translocates carbon and nitrogen, but I'm not sure if a similar response can occur versus from pH. So I think the pH may be more indirect effect on availability of nutrients. So that's probably a long way of avoiding giving you a real answer, but sorry about that. It's not something that I've looked at.
Thanks, Wilfred. Derek, I can see your hand up. Do you have a question?
Yeah, I've probably got a few questions actually, but one of the observations, Wolf, was interesting was when you were talking about bulk density and how the fungi behaves in high bulk density, low bulk density. It actually sounds like the way humans operate. And you've got sparse colonies and they follow preferential pathways, but in the urban areas, there's really dense colonies in smaller spaces. I mean, that's just an observation that seemed very strange, but it could be an innate human behavior, biological behavior. But going back to what struck me when you were talking about the fungus's ability seems to be related to connectability in pore spaces, air pore spaces. So does that mean under things like recent heat waves where the soil's drying out, are we expecting the fungus to rip through, and then next time we'll see a larger explosion? So is there a predictability to this?
Well, I hope there is, and this is one of the scenarios that we will investigate next with our modeling to make our systems more dynamic, because at the moment what we have done is looked at static environmental conditions, and we've not introduced those dynamic changes. When we start doing that, we'll have to consider that when a soil dries, the soil structure will change. So this is not an easy question to answer. The soil will crack, new pores will open up. But strictly, you are correct that for some fungi, if you think back of the later slide, when we have more drought, we would expect more rapid spread. So this is, again, I'm afraid, going to be an that depends answer, because if we would just look at the earlier example I gave, where we looked at air-filled pore space, then you would say, yes, what you just asked me, that is the case. But when we go back to the later slides, where we also considered the diffusion of dissolved organic matter, which in a drier soil will be less, and therefore there will be less available carbon for the fungus to explore. So we've got some contrasting effects that we're working here, and it will lead to some sort of an optimum scenario where fungal growth will be most rapidly. And I'm afraid that at the moment I do not know at what point of wetness that will occur.
I mean, if there's no other ones, I can ask another. I was interested in, have you thought about neutron tomography?
Yes. Just waiting. Yeah. We didn't just think about it, we've actually done it. So and neutron tomography can be done here in the UK, and this will give us a better idea and see different things in soil, so we have a better idea of where water and where organic matter is, but it's also at a different spatial scale. So the advancements of those techniques that we look at now are phenomenal, but I think the biggest advancements are going to be made when we start combining techniques. So the work that's currently undertaken, and we've done it, but also others have done it, is combining X-ray CT with neutron scanning. We also combine X-ray CT with SEM EDX or with nanosims, and it's bringing all those techniques together that will give it an understanding. But yes, there's a recent paper, I think, in the European Journal of Soil Science, which will show you how the two were combined to get a better idea about where organic matter is. The only reservation we'll have to make is that the resolution of X-ray CT is far better than it is for neutron scanning.
Okay, cool.
Are there any other questions? Do feel free to put something in the chat. If you're too shy to say it verbally, feel free. While you're thinking of horrible questions to ask Wilfred, I wonder if I could ask one, take the chair's prerogative. I'm interested in the scale issues that you've been talking about. Can I ask sort of two ends of the spectrum? One is, what are the implications of the findings in terms of carbon sequestration and storage, which we know is very, very important, and the potential for soils to help mitigate climate change in that way? So that's looking at sort of scaling up, if you like, but also if you had a wish list of how smaller you would go, how much of it are we actually missing with the techniques that you've mentioned? And do you think it's valuable if we had the technology, the methods to actually look at even smaller pore spaces that we might be missing now?
Thank you, Jane. These are two interesting and challenging questions. I will start with the last one, which is the resolution. So I can't remember if I mentioned it, but for a typical soil core sample, our resolution is probably about between the 30 and 100 micron, depending a little bit about how we do the scan. So, but 30 microns is certainly possible for a typical soil sample that we currently take in the field. Now 30 microns means that for most soils, we probably still miss about 50% of the pore space. So that can be quite significant what we are overlooking there. And this always raises questions like, what's the value of doing this sort of work? And we have many debates with colleagues about that, and we've also looked into it, because currently we're not able to look at these smaller pores, but we can take a very small part of soil, a much smaller part, and scan at a much better resolution. So we can go sub-micron, which will give us some idea of the pore size distribution at these smaller scales. But still, it will not get us at all of the pore volume. So the way I've looked at it for fungi, so with the Rhizoctonia, so the width of a fungi is probably about 10-15 microns, the internodian length about 100 microns. So when you think about mycelium branching and finding its way through a pore system, it is not surprising that it ends up in the larger pores, because it will be struggling to even get into a smaller pore and really needs to be forced in there. So will it go into much smaller pores? Well, probably not smaller pores than the diameter, so we're not that far off. Secondly, when you think about where water is in pores, that's mostly in the smaller pores rather than in the larger pores. So for most of the situations where we've looked at, the pores that we can see are all air-filled pores, and that's where most of the spread occurs. So we have run some simulations, because we can, of course, do and assume what if we put 50% additional smaller pores into that black pore space. and we look at the effect of that. But this, I think, becomes more relevant when we start looking at bacterial spread, where I will be slightly more worried about that. This is probably the reason why I stick to fungi, because they're more interesting, and I think we're more at an appropriate scale. So there is some hope that we will make some improvements in the future in getting better scans. There's already with dual targets on there to improve the resolution, but I think we'll have to accept that this will always become limiting. The scaling one we're less advanced with, I must say, and this remains a problem. So I think there's a couple of ways by which we can address this. The first one is that we don't always need to scale up what we do. We can start in the field and take our samples and then analyze that, like I showed you for the different tillage systems. So we can make the link with larger scale processes there straight away. When it comes down to carbon modeling, it will be impossible to do this at a national scale or even at a field scale. So it's not possible to run simulations at a pore scale at a national level. And that's not what we should aim for. What we should aim for is to rethink the relationships that are in those models. So currently, the nonlinear relationships that I've shown you are not in the soil carbon models, and they could be easily implemented. So the way that we go with the research in this area is to identify what are the important characteristics at this very small scale that we definitely need to retain to put into our larger scale models. And this is beginning to give us some answers. So from the scans, we can calculate the nearest distance to larger pores. And these sort of factors we've already found out are the key drivers for denitrification into soil. So for various processes, it is the way around that we can identify what are important microscopic characteristics that we need to retain. And a final comment on that is that upscaling is always a question that comes up, but it depends what the process you're interested in. So if your question relates to how microorganisms respond when you have a seed coating and how it subsequently will explore your rhizosphere, you don't have this upscaling problem. The processes happen at a microscopic scale, and therefore what's being done here is directly relevant. So I hope that one of the things we can achieve with this is actually understanding how the rhizosphere functions, get predictions of the rhizosphere, and that will help us to identify what type of organism are easiest to introduce into soil and how that should be done. So for this, we're already working at the right scale. So we're not quite there, but I also think we're not trying to get there. We're trying to follow routes that we've already done. And some of the results work at sort of a sample ring we already take. And to be honest, every measurement happens at that scale, and we tend to use statistical models for scaling up currently to the larger scales. And there's no reason why this can't be done with these sort of simulations.
Okay, thanks, Wilfred. Thank you. Mr. John Chin has his hand up. So, John, your question, please. I think you're on mute, John.
Try again. Is that better? Sorry about that. As the farmer in the room, I'd like to try and tease a bit more out of Wilfred if I can. This is when you have to take a risk, as farmers do, Wilfred, and you have to start guessing what the practical results might be with your scientific knowledge. If I want to try and inoculate and improve the arbuscular mycorrhizal fungi content of my soils, knowing what you know, do I try to pre-inoculate some compost and then spread that on? Do I know what time of year is likely to be best to do this, bearing in mind soil temperature, soil moisture content? Am I more likely to be more successful with a sandy soil or a clay soil, giving the bulk density that might react? Because I've got to take some broad lessons home from this, and I wondered if you could give me any.
Yeah, thank you for that question, John. I need to think a little bit about that. So one of the things I would first say is that when you think back about the colonization of different densities of soil, my first feeling would be that when the soil is a little bit more dense, you get a better spread and therefore colonization of the mycorrhizal fungi. So that would be my first point to bring forward. But having said that, that would only be when you introduce a high amount of inoculum into your soil. So if you have a bit of a more denser material and maybe a bit more of your loamy soil, then probably you need to introduce a higher amount of inoculum in the soil, which will then have a very dense colonization. If you've got a lighter soil, you could probably have a lower inoculum density and rely more on the fungus to spread and colonize the soil that way. So I'm not quite there yet to give you a real answer, John, but just thinking out loud, these are the sort of things that can be obtained from that. But whether you would put fertilizer on there or not, I'm reluctant to give you an answer to that, John. I would need to look a little bit more into that. I'm just trying to think, but we did it with bacteria, because the question then becomes also, if you put organic matter and inoculum in there, does it move out and colonize the soil, or does it make use of the materials that you put in there? And that I would need to look a little bit more, and a little bit more understanding of the behavior of the organism, I'm afraid. So you've reached the limit of my knowledge there, John.
Thank you. Thank you.
I don't believe that for 1 minute, Wilfred, that you've reached the knowledge of your edge of your knowledge. But are there any other questions? I can't see anybody else's hands up. So I think, given the time, if there are no more questions, I think I'll draw this seminar to a close. I'd first of all like to thank everybody for attending, and I hope you've taken away a lot to ruminate on, and I'm sure Wilfred would be delighted to take any questions. If you think of anything after the event, then feel free to contact Wilfred. I'm looking at him to see if he's nodding. He's nodding. Yes, okay, that's good. So I'd like to thank everybody. I'd certainly like to thank Donna and Jess for organising this, and most of all, I'd like to thank my colleague Wilfred for an absolutely fascinating talk. And I work next door to him, and I didn't even know a lot of this stuff. So it's been absolutely fascinating and really insightful. So thank you to everybody. I probably need to give a plug to the next seminar, but I don't know anything. I do know something about it. There's something in the chat. Donna's put something in the chat about the next talk, which will be on the 13th of October at 4 p.m. And there is somebody talking about phenotyping and data processing at 4 o'clock on the 13th of October. So without further ado, I'll bring this seminar to an end and thank everybody for their contributions, and have a good rest of the week, everyone. Thanks a lot. Thank you.